Hello guys, today we're going to be solving IB Business Management May 2023 paper 1 and this is time zone 1. So yeah, so we're going to be solving and explaining the answers. So for the first question, the micrograph shows two types of blood cell. Okay, so we know that this is a blood cell, right? What determines the difference between the two types of cells? Okay, so they told us there's two types of cells. So the first answer says different number of mitotic cycles, different expression of some genes, and reaction to oxygen of red blood cells and reaction to antigens of white blood cells. It's B because two types means that they just differentiated into different types with different roles. Okay. Because if there are two types of blood cells in the same body, then there's no different number of mitotic cycles. You know, that does not differentiate them. Or reaction of oxygen of reaction to oxygen of red blood cells. Again, no, because usually the red, like the alveoli just takes in the oxygen and releases the carbon dioxide. So this has nothing to do with it. And reaction to antigens of white blood cell, again, that has nothing to do with it. I mean, if you want to think of it another way, which is kind of simpler, and if it really makes your life easier, is that in IB biology, I give this tip to all my students, is that we go from 1.1 to 6.6 in SL, which is what we're solving. We're solving SL papers. So this is technically 1.1, right? So the 1.1 is about differentiation and expression of genes, and this is how genes are different because they express genes. So it is B. Our answer is B because this is the only one actually related to 1.1. The rest are not related. Mitotic cycles actually related to 1.6. And there's no such thing as cells being different because of mitotic cycles. That's just false. And then you have like here, you have here, that's 1.6. Oxygen reaction of oxygen to red blood cells. And these two just they're like in chapter 6. So we don't really need to like get there. So then the second question says, um, what is the difference between eukaryotic cells and prokaryotic cells? This is a really important question. It's something really important. Again, this is 1.0. You see the pattern here? Eukaryotic, prokaryotic. So cell walls are only found in prokaryotes, which is really, really, really false because we know that, for example, eukaryotes, which are plants, have cell walls. And this is actually found in plants. And plants are, well, not all of them are prokaryotes, but some of them are eukaryotes. So this is false. Um, Naked DNA with histones only found in prokaryotes, and this is false because histones are actually in eukaryotic chromosomes. Okay, they're found in eukaryotic chromosomes. They're not in prokaryotic chromosomes. Compartmentalization is only found in eukaryotes. This is true actually. So our answer is probably C. Cilia and flagella are only found in prokaryotes. This is false. Flagella is actually a tail that helps organisms move, and cilia are like hair-like structures, and they're like in the body, and they had like locomotion. So no, because sometimes flagella, for example, is found in eukaryotes. So C is a right answer. So yeah, for example, I just want to talk about eukaryotic and prokaryotic organisms, but I'm scared you won't have time for the video. But basically, eukaryotic and prokaryotic chromosomes are completely different, and you actually should study that because they come in exam questions. So the next question, cytochrome reductase is a transmembrane protein which acts as a H plus channel in respiration, which characteristic allows cytochrome reductase to be a transmembrane protein. So it's hydrophilic except for the central core, which is hydrophobic channel. This is false. Okay, because it actually can't just have a hydrophilic part and then the central core be hydrophobic because it just won't make sense for the protein to transport. It has a hydrophobic regions embedded in the membrane, um, and hydrophilic regions exposed on both sides of the membrane. That is true, okay, because the hydrophobic regions are in the membrane because they can't embed in the membrane because they're like against water. So because hydrophobic is basically water hating, okay? And hydrophilic region exposed on both sides of the membrane. Okay. um, because they are water loving. Okay. It has a hydrophobic central core, um, acting as a channel, and hydrophilic a region embedded in the membrane. That's not true. It's hydrophobic except for the part in contact with the phospholipid membrane. That's not true. So moving on. So for the next question, onion epidermis was placed in pure water and observed with light microscope using high magnification. What would happen if these cells were transferred to a hypertonic solution? Okay, so basically, let's say hypertonic is basically when the concentration is higher, hypotonic is when the concentration is lower. For example, let's say, I'm just going to give you examples to explain this lesson in a short way. So this, let's say our solution is 0, right? The solution is 0.2 concentration, this solution, or let's make this 0.1, never mind. 0.1, this is 0, hypotonic, isotonic, and hypertonic. Okay, I hope you got me. So what happens in hypertonic solutions is that it, just make it another color, decreases in size, shrinks, increases in size, swells, okay, or lice. This was lice is also a word you can use, and here just stays the same. So what would happen if it's placed in a hypertonic solution? So here we directly found our answer just by reviewing the lesson, just remembering it during the exam. Cell would gain mass, no, because this would be for a hypotonic solution, hypotonic. Cells would take in water by osmosis and swell. Again, this is a hypertonic solution, not a hypotonic solution. Cells would burst open, releasing the content. Again, this would only happen when it would burst and lies in a hypertonic solution. Cells would detach from their walls at some points. D is our answer. Okay, because this would happen because the solution is hypertonic, so it ends up shrinking and it will end up detaching from the walls. Anyways, these three are completely impossible, so D is our only answer by elimination. Question 5. Which evidence for the endosymbiotic theory in eukaryotic cells? So again, quick review of the endosymbiotic theory. It's about mitochondria and the chloroplast, and how these two basically they are prokaryotes which were engulfed by eukaryotes in order to work for them. They're basically the same size as prokaryotes. They both produce by binary fission, and they have 70S ribosomes, okay, and they have similar DNA to the prokaryotes. So for the first answer, it has mitochondrion with DNA. This is technically correct. Golgi complex in cytoplasm has nothing to do with endosymbiotic theory, sorry. Singular nuclear membrane, again, no. Ribosomes in the cytoplasm, no. The answer is A. Really simple. What's the example of catabolism? Okay, so let's do this question. Maltose produced from glucose molecules? No, because again, this would require two glucose molecules to produce maltose molecules. Again, this is anabolism. Anabolism is when A plus B leads to C. Okay, whereas catabolism is something else. It's basically C equals to A plus B. And starch produced from monosaccharides. Again, no, because two monosaccharides produce disaccharides and produce polysaccharides. So this is a no, this is a no. Peptides produced by protein digestion, yes, okay, because the protein is broken down to a peptide. This is after digestion. So breaking down takes place. So this is our question. And then D is ethanol and fatty acids produced by triglyceride digestion. No, our answer is C in this case. So 7. So 7, they're asking us which molecular diagram shows part of the cellulose molecule. So for A, we actually can see the cellulose molecule because we can see the 1,6 branching. Okay, so the 1,6 linkage. We can see it here. So we can say it's correct. B, no, it just doesn't work because there's no 1,6 linkage. This is a 1,4 linkage. This doesn't even have a glucose molecule. This is not a starch. So this is not a carbohydrate, I mean. Carbohydrate, I guess. So no, wrong. And this is wrong because these molecules, they're not carbohydrates, both of them, so our answer is A. So now we have 8. The table shows approximately energy stores in a man with an average mass. So we have like the amount of, let's say, carbohydrates, lipids, proteins. which can be concluded for them died. So okay, so we have to read and then look, because if you keep looking and then you read, you're just gonna end up wasting time. It's gonna end up taking you more time. So what can be concluded from the data? The brain contains no short-term energy. Okay, so now we know that for short-term energy, we have carbohydrates, right? They provide short-term energy. And the brain has 30 kJ of short-term energy, so that is false. B, the liver contains less long-term than short-term energy. So let's look at the liver. So we know that the liver is long-term and 2000 is greater than 1700. So again, this is false. The adipose tissue provides the most long-term energy storage. So let's look lipids. Yes, 56,000 is greater than 2000 and greater than 0. So C is correct. Carbohydrates provide more energy per grams than lipids or proteins. And again, this is false because just look at the data. So C is our answer. Now for 9. Hemoglobin is a protein made up of 2 alpha and 2 beta polypeptide chains. In sickle cell anemia, a mutation causes one glutamic acid in beta chain to be replaced by valine, as shown in the image. Okay, so here they were nice enough to actually explain to us, even though this is required for you to know in the syllabus, they were actually kind enough to explain. So how does this mutation in hemoglobin cause sickle cell anemia? So if you don't sickle cell anemia is that when less red blood cells are produced. So here it prevents the beta chains from forming a protein. That is false, does not do that. It replaces the amino acid with a fatty acid in the beta chain. No, it replaces it with something else. But this is actually because when the replacement happens, it can either stay the same, it can be better, it could be worse. In this case, this is a worse, but it cannot replace it with a fatty acid, so this does not work. It changes the three-dimensional conformation of hemoglobin. Exactly, this is what happens. It ends up changing it. So hemoglobin does not work the same. So this is our answer. The polypeptide produced in sickle hemoglobin is shorter than normal hemoglobin. No, it's again the conformation of the protein will end up changing, because again we do know that it is a protein. Hemoglobin is a protein. So what happens is that the 3D conformation of the protein will end up changing, and this is what happens. C is our answer. Um, for 10, sorbitol is oxidized to sorbose by the enzyme sorbitol dehydrogenase. The conversion rate was measured by monitoring sorbose concentrations. The graph shows the activity of sorbitol dehydrogenase at different pH levels. So here we have the pH, here we have the conversion rate. So you can just see it just increase and, decrease. But so we can kind of get this around optimum. But anyways, what can be deduced from the results? There is no conversion of sorbitol to sorbose at pH 10. Actually, there is kind of one. It's not at 0, so this is false. At least 10% of the product is transformed to substrate at pH 9. This is actually not completely true, because they're not talking about the product, they just talk about the conversion rate, at which rate it um transformed. So this is not really accurate. The pH affects the rate of activity of the enzyme sorbitol dehydrogenase, and yes, we can tell that we have optimum, and then here they go below the optimum, and it kind of starts to like not work as much, and maybe even like what's it called, the protein begins to like deform, like the 3D conformation is changing. So this is true. The amount of sorbus produced is not affected by the substrate information or substrate concentration. That's not true, but because basically it is, because the more substrate concentration there is, the more the reaction is going to be stronger. So this is false. D is false. So 11. How do DNA and messenger RNA, which is basically mRNA molecules, differ in eukaryotes? Okay, so basically we have DNA versus RNA to a certain extent. That's what they're asking us. mRNA is twice the size of DNA and is formed from 4 bases. This is false. It's not twice the size of DNA. This is not true. DNA contains ribose, but mRNA contains deoxyribose. This is wrong. DNA is deoxyribose, okay? Sugar, where RNA is just ribose sugar. So they actually flip them. So this is just the opposite. mRNA has the same basis as DNA, but contains ribose. Okay, so the ribose part is false. It's actually true, sorry. The part that's wrong is that mRNA has uracil, whereas DNA has thymine. So this is wrong. DNA has two strands, but mRNA has one strand. That is correct. So our answer is 11. So this is just down to memorization. It has no analysis, just has to see whether you know your lesson or not. There's no analysis, there's no thinking required. And we don't have the image for this one, so we're just gonna skip it. Yeah. So we don't have this, so we're just gonna skip it. 13. What is the consequence of anaerobic cell respiration in muscles? So Lord G, okay, so it's an anaerobic, does not require oxygen. Muscles, okay. Lactate usually is produced, but anyways. Large yield of readily available ATP in ethanol production. No, ethanol production is false. This is basically in bacteria, not for muscle. So A is wrong. Small yield of ATP, which is correct because anaerobic is large yield of ATP. Aerobic, small yield of ATP, okay? And lactate accumulation, like I said, so B is correct. Large yield of ATP, this is false. And production of carbon dioxide and lactate acid, no. Small yield of ATP that's not readily available and carbon dioxide production is wrong. It's wrong, so these two are wrong. So our answer is B. Okay, so scientists sequence the genes in each chromosome of chimpanzees and humans. The graph shows the mean divergence between the genes of these species by chromosomes. What can be deduced from this data? Autosomes are more similar than Y chromosomes, and you can actually tell because look at the difference. Like the best fit line, let's say if you just make a best fit line, you can just tell. Just give me a second, this is taking so much time, but basically you can just tell that when you reach the X and the Y, because these are all autosomes, or basically, and these are gametes. They're the chromosomes that basically determine the sex. So this is technically correct. There is the same number of chromosomes in humans and chimpanzees. No, there isn't. And yeah, because humans have 23 chromosomes. So they have, this would be 22, and then here would have XY. So yeah. Humans are more closely related to chimpanzee than to other species. No, we don't have any data discussing any other species. Smaller chromosomes are more similar to larger chromosomes. No, this doesn't have actually the gametes are actually smaller than the autosomes, so this is false. So A is our answer. For the next question, non-destruction of chromosome causes trisomy. So yeah, 21 causes trisomy. It's called trisomy 21. It's basically Down syndrome. What can be said about the incidence of non-destruction in chromosome 21? It increases greatly with the maternal age after 30 years. This is actually true, and there's actually a graph that discusses this in the book and talks about it. You can visit it in BioNinja. I think it's section 3.4 or 3.3. No, it's actually 3.4, I think. I don't remember, but it's one of it's in topic 3 definitely. Chromosome very one autonomic always suffers on the dysfunction. No, this only happens when there's trisomy 20 that causes trisomy 21. Not everyone has trisomy 21. Very young mothers have no chance of this disjunction. They do, but it's just slower. Just slower. So this is false. Like this, the technicality of the words, the grammar is wrong. There's no relation between age and probability of disjunction. No, that is not true because it usually decreases over time, and especially at the age of 37, 38 to 39, just it increases a lot. So this is not true. For what reasons do gametes only contain one allele of genes? To prohibit inbreeding in a population? No. Haploid cells contain only one set of chromosomes. This is true because haploid is N, diploid is 2N. Okay. The two alleles of a gene are separated during mitosis. No. Crossing over will always produce one gene of allele. No. So this is it for part 1, guys. I will do to part 2 right after this video, but I just don't want them to be this long. So thank you for watching. Subscribe, like if you want more questions. If you have a question, if you have a specific paper you want me to solve, then tell me. Please like and subscribe so I can continue making more videos for you guys, and join my biology Google Classroom. I'm going to be putting the link down below soon. Thank you so much for watching, guys. Bye bye. And part 2 will just be coming out in a while, just a small while after this. I'm just going to film it right after. Thank you guys for watching. Bye bye.